The vascular endothelium forms a robust barrier between tissues and their blood supply. Yet many blood cells must enter or exit the circulation, crossing the endothelium in a process known as diapedesis or transendothelial migration. As leukocytes pass through or in between endothelial cells, they create small holes in the endothelial monolayer, which, as Christopher Carman from Harvard Medical School in Boston explains, must quickly be closed so that the endothelium can maintain its barrier function.
There's actually been remarkably little done to understand how the endothelial cells are able to close up these discontinuities at the completion of these migration events. So we were at the starting point of wanting to ask the question at a cellular and molecular level, what are the dynamics for closing these discontinuities, or as we like to consider, healing these micro wounds. So we set up in vitro models to do that.
Carman and colleagues, led by Roberta Martinelli and Masataka Kame, initially looked at endothelial monolayers expressing actin GFP and a fluorescent plasma membrane marker. Leukocytes passing through these cells left small transcellular pores in their wake, and, as Martinelli explains, the researchers expected to see actin assemble all around these holes and draw them shut through the same process that closes holes in the membrane of Xenopus oocytes.
However, what we actually observe is that every transmigratory event closes through a largely asymmetric process. And we were able to observe a dynamic burst of actin that formed on one side of the microwound and then rapidly moved across to close it.
Single actin bursts could close multiple transcellular pores and appeared to be membrane protrusions similar to the lamellipodia that form at the leading edges of migrating cells. But did these pore-closing protrusions form at the dorsal or ventral surface of the endothelial monolayer?
To answer this question, we decided to employ turf microscopy, IRM, and spinning disk confocal microscopy, and we showed that in every case, the lamellipodia structure was propagating underneath the endothelium, on the ventral side. And we thought that it might allow integrin matrix adhesion to participate in the closure and in the formation of the ventral lamellipodia. So, in fact, we found that the ventral lamellipodia were enriched in alpha-5 beta-1 and alpha-V beta-3 integrins, and acute addition of function-blocking antibodies to this molecule were able to cause the arrest and retraction of the ventral lamellipodia and a failure for the microwounds to close.
Ventral lamellipodia also formed when leukocytes migrated between endothelial cells, helping to close the paracellular gaps and re-establish intercellular adherens junctions. And similar protrusions formed in epithelial cells to close the holes created by transmigrating leukocytes. Martinelli et al. Therefore began to analyse these ventral lamellipodia in more detail.
We undertook a careful empirical analysis of literally hundreds of these closure events. First of all, they could form at a significant distance from the edge of a discontinuity, on average about 5 microns, but sometimes much more. We also noticed that those initiation sites tended to be pre-existing actin fibers that we termed a parent filament, which, though not necessarily part of the discontinuity, were invariably in some way distorted or even broken during the transmigration event.
Actin filaments assembled around these parental fibers, forming ventral lamellipodia that propagated towards microwounds regardless of their relative position in the cell.
Another striking aspect was we could often see them changing direction in the course of their migration. And a given ventral lamellipodia that's been initiated to close one discontinuity could then continue on and even change its direction to move on to close subsequent pores or gaps.
All of these observations suggested that the formation and propagation of ventral lamellipodia might be regulated by mechanical signals. For example, transmigrating leukocytes could perturb the tension in endothelial monolayers by disrupting cell-cell and cell matrix adhesions, thereby initiating ventral lamellipodia and micro-wound closure. To examine the role of mechanical signaling, Martinelli et al. Used a tungsten probe to mechanically wound endothelial cells independently of leukocyte migration.
When we break the local actin fibers, tension is released and you have a viscoelastic recoil that causes a rapid expansion of the wound, typically to about 30 or so microns. Following this initial expansion, rather than a single ventral lamellipodia, we mobilized sometimes 10 or 30 of these structures around the periphery of the mechanical microwound, which then migrated and collectively restored these larger microwounds.
The viscoelastic recoil induced by mechanical wounding also seemed to be transmitted through intercellular adhesions to initiate ventral lamellipodia in neighbouring non-wounded cells. To test the idea that a sudden loss of tension upon wounding induces lamellipodia, Martinelli et al. Lowered initial tension levels by pre-treating endothelial cells with drugs that block tension development. They then examined the cell's response to mechanical wounding.
In these cases, we were able to show that the presence of the drug resulted in decreased rate of wound expansion, so the tension was reduced, and as a consequence of this, there is a decreased rate of recovery of the wound.
Lowering initial tension levels therefore blunts the wound response. The researchers then examined whether a sudden loss of tension was sufficient to induce lamellipodia in the absence of a wound.
We basically would culture a monolayer of cells on a pre-stretched silicon membrane and impose an acute 10% de-stretch, or essentially a compression, in a uniaxial direction. And very satisfyingly, within tens or 30 seconds or so of the compression, the endothelial cells generated bursts of what looked to be a very analogous structures to the ventral lamellipodia along the edges of cells that were parallel to the contraction.
This suggests that a sudden loss of tension induces wound-repairing lamellipodia. But what molecules are involved in this process? Martinelli et al. Examined the role of RAC1, a GTPase that induces lamellipodia and is known to be activated by tension release.
Acute treatment with the RAC1 inhibitor or the presence of the dominant negative RAC results in a reduction of ventral lamellipodia formation and blockade of pore closure. Additionally, if we washed out the drug, we had a rapid and synchronized mobilization of multiple ventral lamellipodia, meaning that this effect is totally reversible.
An inhibitor of the RAC-regulated actin-nucleating ARP2/3 complex also blocked ventral lamellipodia and micro-wound healing, as did a dominant negative version of the RAC effector cortactin. Indeed, several RAC effectors accumulated in ventral lamellipodia, including the NADPH oxidase subunit P47-FOX, which generates reactive oxygen species such as hydrogen peroxide.
We transfected the cells with a H2O2 selective GFP-based biosensor that we targeted to the membrane, and we were able to show that there is production of H2O2 in the ventral lamellipodia during transendothelial migration and mechanical microwounding. And if we block this production, there is an inhibition of the closure of the microwound.
The researchers now want to investigate how hydrogen peroxide stimulates lamellipodia formation and to determine how a loss of tension is sensed and relayed to RAC and its effectors to initiate wound repair. But Carman and colleagues are also interested in the wider implications of their work.
So the existing paradigms for mechanobiology really focus on how force loading on a mechanosensitive element positively modulates responses. And what our findings basically show is physiologically relevant settings whereby tension loss is capable of positively signaling a recuperative response. So this gives us a broader way to think about how mechanobiology may work and how cells are informed by their mechanical environment.
Similar responses to tension loss could, for example, be involved in coordinating collective cell migration. But in the meantime, you can learn more about how tension loss induces ventral lamellipodia and the repair of endothelial microwounds in the paper by Martinelli et al., published in the April 29, 2013 issue of the Journal of Cell Biology.